I think it could really improve our ability to not only study epilepsy in clinical trials and do those better, but many other brain disorders. Because the brain is an electrical organ, and even in diseases like Alzheimer's, you have spiking and things like that. And ADHD, you have spiking but not seizures. So if we could get this to work and we could see the brain at that millimeter resolution of all these different metabolites, I think that would be tremendously powerful for many diseases. So that's something we're actively working on. So I want to finish. Am I doing okay? A couple more slides. Putting it all together and talking about where's the future, where are we going with all of this. We've talked about our database and creating integrated data sets that allows us to really ask questions about anything in relation to anything. And you can form these hypotheses and then test the hypotheses between, say, electrical activity and genes like we did. Or you could just create an entire interactome of data and mine it to see what jumps out at you, sort of the low-hanging fruit approach across all the different data by finding relationships between all of these different measures, because we try to get every measure with some sort of a numerical value on it so we can actually do a comparison. So here's an example. This is a particular neuron type 4 that's predicted. Goes up in these samples, goes down. This happens to be a nuclear protein that has the same pattern of expression, just a Pearson correlation coefficient. We can give it a number and give it a strength. Here's another example of that same neuron population, but this is a long non-coding RNA, an RNA that doesn't even make proteins. And it has a beautiful mirror, an opposite Pearson correlation coefficient. And this is so you can basically, and you could do any kind of mathematical relationship of all the data to everything and ask, well, then what does it look like? It looks something like this. So this is going back to Cytoscape, taking all the data in the matrix from different types of RNA, coding, non-coding, metabolites as the little red squares, different protein fractions, membrane, nuclear cytosolic, cell types. What medication does the patient take? What is the age of the patient? What is the frequency or type or semiology of the seizures, the slope of the spikes? And then create a relational map. And then things that cluster more together or groups are things that we can then go and mine and explore. So here's an example of a cluster down here. This cluster actually happens to have back to our friend, the red blood cell. Look at the proteins and triangles that go with this. HLA, IgG, hemoglobins, things that we know are in blood from the proteomic studies. So very quickly when we start looking at these relationships, things kind of make sense that it's working, that we find blood and we find blood proteins that come together. So it kind of makes sense. But there's all these unknown things like genes and metabolites that we may not have expected to be in this cluster that is basically giving us the sort of discovery machine of new things that we didn't think about. A medication happened to pop into this group. It's a medication called Vimpat or lacosamide. It's one of the seizure medicines. One of the side effects can be anemia. Maybe there's some reason, and we can actually see what proteins this drug interacts with to actually describe this and go back and maybe sell it to the drug company and say, hey, we got some interesting stuff. You want to work with us? And we've been trying to do that. Here's another cluster of variables. These are age-dependent variables. So we have patients all the way from 6 months of age to 50 years of age. So there are some variables that cluster or that correlate with age. And this medication, vigabatrin, comes in this cluster because it's only given to babies who have something called infantile spasms. So not surprisingly, of course, this drug is an age-dependent. But look at all the other age-dependent variables in here. And this data set is not statistically powered as much as we like. We need to add more patients, more genes, more proteins, and we keep growing and building it. And so one of the challenges of these type of integrative data sets, we built this in an Oracle, very rigid data structure, and adding a field is like pulling many teeth when you want to put something new or create some new, or the structure of our gene annotations change, everything changes. So we need to build a new model and create what we call a big data dream. And this again is work being done by Neil Barus with Biswajit as well, trying to not just put data that we enter, but take the entire electronic health record and basically feed that data together with the entire data from an imaging server. Every imaging study at each time point that a patient had, histology server. So if you have a piece of tissue, say here, at this location, links to that electrical activity, links to that histology on a histology server, links to a tissue inventory server, and then links to all of our omic data and also to external servers. And then we create analytics that allow us to really, in real time, mine on a web-based interface so that we can ask any question we want and share this data with the world. So big dreams, But I think it's really possible. It's something that we're pushing for with the help of our CCTS here. I think it's something that we could even share across different centers that do this type of research. And that's all I have to say. I do want to say that there's this type of work as a core group of people that do a majority of the work, but lots of collaborators in pretty much every field you can imagine, including engineering, neurology, neurosurgery, pediatric neurology, and industry. So I want to thank you for your attention, and I'll field any questions you have.
That was really fabulous. I hope you saw he illustrated what I talked about time series and also clustering coming into patterns. So patterns sometimes can be underrated. But it's helping to understand how we take a large set of behaviors and information, and we're narrowing down on things. Really interesting. So this is the way it's going to work. Anybody who has questions, please just raise your hand, and I'll bring you the microphone so that we can hear your question and or comments or discussion. So anybody have something in mind right away? Yeah, Bill.
Great talk, very fascinating and in-depth. How many of these techniques are available to be applied to other diseases? You briefly mentioned Alzheimer's and some other things. Then would this help those diseases also?
So the goal of moving forward with this is not to create an epilepsy-only specific way of doing this. So once we build a way to interact the electronic medical record with an imaging server and a histology server and a tissue inventory, we can put any disease in there we want. So the goal is there are going to be modules within this data structure that allow us to apply this across any disease. It can even be outside the brain. But there's also going to be disease specific, because we don't do electrical recordings in patients with Alzheimer's disease. But all the other things we would be interested in doing. So the idea is to build a data structure that allows us to apply this to any disease you want to study. But you need to keep track of the three-dimensional brain space, where the tissue came from, where the genes were, where the imaging is. Because going back to the imaging is going to give us many biomarkers, many tools to understand, to diagnose, and treat disease.
Thank you. That was Professor Zangler, by the way, another friend of mine on the team. You want to make some comments, Doctor?
I've got a question. Regarding the kind of research that we might discover now that we've got this data set pulled together, it feels like obviously the quest for biomarkers, you've got a lot of information on tissue, you've got a lot of on genomics. Is protein biomarkers the unmined part of this research, do you think, that's going to be available?
Right. So I didn't show you data, but we do have, as part of that interactome, the triangles were proteins. Interesting, it's a very separate set of biomarkers. When people have looked at what's called proteomics and genomics, they often don't relate to each other. So it's a separate, and like metabolites. So absolutely, I mean, proteins are far more complex than genes. Genes are kind of simple. You got a gene, it goes up, it goes down. There can be changes in what's called splicing of the gene, but it's the gene. The proteins have shapes and folds and modifications and phosphorylations. There's infinite possibilities. So absolutely. But again, the idea of the big data is, as our technologies improve to enter new protein data and new gene data in, our data structure has to be able to accommodate those new models and modes. So absolutely proteins. That was my PhD, which was in membrane biochemistry. So proteins are near and dear to me.
Hi, Tanya Berger-Wolf, professor of computer science here. So I appreciate very much that you try to validate in vivo and experimentally most of the hypotheses, every hypothesis that comes out of the big data mining. Because one of the biggest problems with these analytical techniques, including network analysis and that you brought in, is that you showcased here, is that we really have no way how to evaluate the statistical significance of pretty much anything that comes out of it. And particularly when we're talking about 5 patients, or even 15, or even 50, it's still not enough quite often for a lot of, to give any kind of support, statistical significance values for the results that come out of this kind of analysis. For example, with cancer, which is very much related in Cytoscape, they had 128 patients with ovarian cancer with 128 different profiles. And it's only putting that together that they have any kind of notion of the network of pathways, metabolic pathways, and, the gene regulatory
Right.
network that comes out of it.
So part of what gives statistical power is the internally controlled experimental design, because you're really only asking within the same tissue an area that has a difference that you can quantify, whether it's histology or whether it's the electrical activity. That's what allows us to get, I mean, our raw p-values are generally 10 to the minus 20. So these are very, very robust changes. You don't need large N. So once you find something using this approach, then you can go do PCR and do larger series to validate and spend less time using large series to identify populations of genes. So I think that helps with lower populations get those pathways. But again, I don't really believe anything we do until we use a different methodology and a different system and go from genes to proteins and find the pathway is validated. Because that's not going to happen by chance. So by going at the different modalities, I think, and the internal control model gives us a lot more power to find things. And you're right, we say we found it in 5 patients, then we looked at 100 patients, we found the same thing. But we didn't have to look at 43,000 things in 100 patients, we just looked at the few things that we were interested in.
That was a great question. I think, too, that there may be some new validation models coming out of some of this. And that's kind of what I was hinting at the beginning, that it's time for some new vision, for analytics. Interesting that you bring up about cancer. I just received this in the mail today, the Economist cover story, new cures for cancer. So everybody's on the same page here. It's kind of interesting.
Yeah.
I'm sure it's more questions. There are a lot of questions in my mind about control. You know, who has access to this data? It's scary stuff. Yeah.
It's all de-identified.
Oh, but once you have genetics.
In your presentation, you mentioned the morning after pill for preventative epilepsy. With all the research you've done recently, where do you see the future of anticonvulsants for people who already have epilepsy in the next 5 years or so?
Well, if you've looked at the number of anticonvulsant drugs, say over the last 30 or 40 years, 30 or 40 years ago, we had 2 or 3 drugs. We had bromides, then we had phenobarbital, then phenytoin. There's been an exponential growth. We have like 30 drugs right now. And the problem is they're all developed the same way on the same animal models, and they all pretty much work in about 2/3 of the patients and fail in about 1/3 of the patients. So I kind of think that the way we've approached drug development so far has kind of plateaued in these anticonvulsants. And we need to think about new pathways and new things we're going after besides just blocking seizures. So all this activity that we're seeing between the seizures is not normal and it's non-random. And looking at it allows us to start thinking about other types of targets to go after, so we're not just making another anticonvulsant that only works in 2/3 of the patients. So the idea of the morning after pill is really disease modification. How do we change the network? How do we change the tissue in some way so that you don't either develop epilepsy, or could we take those neurons that we discovered in those microlesions and normalize them, get them to regrow and sprout so that they create the inhibition that they no longer have from the epilepsy and produce more normal brain again. So there's a lot we can do.
We have another question over here.
Yeah.
So I have 2 questions since I'm from the engineering background. My questions are strictly about model building. Firstly, you said you sort of converted raw data into processable data.
To what?
The white noise raw data, you sort of converted that into data that can be processed.
Right.
So can you just give us any idea about how that is done? And secondly, while processing the data, what sort of approach that you had, like a deep learning approach about the model?
So,
Classification problem.
so the first thing we did was try to create a MATLAB script that says this is a spike, this is a sharp wave on an EEG, and this is not, okay? And people have been working on this for years with all kinds of algorithms. But again, what we did is we created an algorithm, and it seemed to work pretty well. And what's our comparison? How do we validate? Well, right now, the only validation we have is a human person like me who's trained to read EEGs, and I say that's a spike and that's not a spike. Unfortunately, that's the best we have. So we got 3 different human reviewers to say what's a spike and what's not a spike, and then we ran our algorithm. And we look at the concordance rates between the different reviewers and between the algorithm. Now, the concordance rate between the 3 different reviewers was about 5%. It's terrible. So it tells you what we have to work with. But if you look at the concordance between our algorithm and any one individual reviewer, it's much higher. So we do need standard methods. Whether the method that we used, that uses a very simple filtering model, is the best way, may or may not be. But it clearly works better than our traditional way of letting people read the EEGs. In terms of some of the other models, I mean, we can go into some of the details. I probably can't. BizWG can explain it better than I can on some of these DDTF functions and so forth. But everything we do, we have to validate and test, and we have to look at and get somebody to say, I think this spike's first, that one's second, that one's third. Somebody has to actually look at it. And I think that's one of the challenges. You need to actually have some, when you're developing a script, an algorithm to quantify something, you need to have in your experimental plan how you're going to test it, how you're going to take a new data set and let somebody or some other method do it to say whether this method's better or not. So there's no perfect method, but having numbers, the scary thing is most of the time these surgeries are done, there's no numbers. There's just a human person saying cut here because I see spikes here and seizures there. That's the truth.
We have another question back here.
I had 2 questions. The first was, I was wondering whether the inhibitory pill, like, you said it like, kind of like makes you dumb, but is that only temporary while you're in the intensive care, or like it like makes you more dumb forever? And my second question was more about